Hello everyone, I am Vinci, Assistant Professor, Department of Botany, Holy Cross College, Nargovil. Now I am going to discuss the topic respiration. Respiration is a process of biological oxidation where oxygen is utilized and carbon dioxide released for the purpose of releasing energy. All living organisms, whether plants or animals, require energy to drive life activities. Solar energy trapped by green plants during photosynthesis are transformed into one form or the other is released and liberated by oxidative catabolism. This process is called respiration. In respiration, many organic compounds are break down to carbon dioxide and water. The energy liberated is made available to drive life processes and many intermediate are produced which serve as raw material for other metabolic processes. The leaves of plants have tiny pores on the surface which are called stomata. The exchange of air gases in the leaves during respiration takes place through stomata. Oxygen from the air enters into the leaf through stomata and reaches all the cells by the process of diffusion. This oxygen is used in respiration in cells of the leaf. The carbon dioxide produced during diffuses out from the leaf into the air through same stomata. The roots of plants are under the ground, but root cells are also need oxygen to carry out respiration and release energy for their own use. The root of plants uptake air from the spaces between the soil particle. Root hairs are in contact with the air in the soil particle. Oxygen from the air in the soil particle diffuse into root hairs and reach all the cells of the root where it is utilized in respiration. Carbon dioxide produced in the cells of the root during respiration goes out through the same root hair by the process of diffusion. Plant cells are built of mainly organic substances. Some of them are structural because they become part of the cell wall and protoplasmic machinery. Other organic substances serve as reserve food. The reserve food materials take part in the metabolic activity. A respiratory substrate is an organic substance which can be degraded to produce energy, which is required for various activities of the cell. The various respiratory substrate in respirable material found in plants are carbohydrate, fat, organic acid, and protein. Degradation of organic food for the purpose of releasing energy can occur with or without the participation of oxygen. Hence, respiration can be classified into two types, namely aerobic and anaerobic respiration. Aerobic respiration Aerobic respiration takes place in the presence of oxygen and the respiratory substrate gets completely oxidized to carbon dioxide and water as end product. This type of respiration is of the common occurrence and it is often used as a synonym of respiration. When the term respiration is used without qualification, it denotes aerobic respiration. Anaerobic respiration, it takes place in the absence of oxygen and the respiratory substrate is incompletely oxidized. Some other compounds are also formed in addition to carbon dioxide. This type of respiration is of rare occurrence but common among microorganisms like yeast. Glycolysis comes from a merger of two Greek words, glyc and lysis. Glyc means feed, lysis means breaking down. It is also known as Embden-Meyerhof-Parnas pathway or EMB pathway. It is taking place in the cytoplasm and it does not require oxygen. Hence, glycolysis is an anaerobic process. Glucose is first converted into glucose 6-phosphate by the reaction with the ATP. This reaction is irreversible and it is catalyzed by the enzyme hexokinase and require Mg double plus as cofactor. Step 2. Glucose 6-phosphate is then isomerized to fructose 6-phosphate by the action of the enzyme phosphoglucoisomerase. This reaction is a reversible reaction. Fructose 6-phosphate is further phosphorylated to form fructose 1,6-diphosphate at the expense of another ATP molecule. This reaction is irreversible and catalyzed by the enzyme phosphofructokinase. Step 4. Fructose 1,6-diphosphate is cleaved into two molecules of 3-carbon compound in the presence of enzyme aldolase. The two 3-carbon compound are formed are 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. These two compounds are interconvertible and it is catalyzed by the enzyme phosphotriose isomerase. In this step, 3-phosphoglyceraldehyde is converted into 1,3-diphosphoglyceric acid by the addition of 1 phosphoric acid. This oxidative reaction is catalyzed by the enzyme phosphoglyceraldehyde dehydrogenase. In the next reaction, 1,3-diphosphoglyceric acid react with ADP and loses a phosphate group forming 3-phosphoglyceric acid. This reaction is catalyzed by the enzyme phosphoglyceric kinase. This is the first site of ATP synthesis when one substrate is converted to the other form. ATP synthesis occurs. Hence, this type of ATP synthesis is known as substrate level phosphorylation. Step 7. In the presence of enzyme phosphoglyceromutase, 3-phosphoglyceric acid is converted into 2-phosphoglyceric acid. For this reaction, Mg++ acts as cofactor. Dehydration occurs in 2-phosphoglyceric acid by the enzyme enolase to form phosphoenol pyruvic acid. Mg++ is the cofactor for this reaction. The final step of glycolysis is the conversion of phosphophenol pyruvic acid into pyruvic acid by the enzyme pyruvic kinase. Phosphate group is removed from phosphoenol pyruvic acid and it is reacted with the ADP and form ATP. This is the second and last site of ATP synthesis in this pathway. One molecule of glucose produces 2 net ATP. 2 ATP were used at the beginning, 4 ATP were produced through substrate level phosphorylation, 2 molecule of NADH, 2 hydrogen ion and 2 molecule of pyruvate.